I'm Jian Li from Monash University in Melbourne, Australia. This webinar is co-hosted by Anti-infective Pharmacology Working Group of the ISAC, the International Society of Antimicrobial Chemotherapy, and the China Agricultural University. So before I start, I just want to say a few words about these two hosts. So ISAC is an international society on antimicrobial chemotherapy, aims to advance the education and science of therapy infection. ISAC has a worldwide membership with a very strong partnerships with 93 member societies. So I think if you are interested to join our society, ISAC, please do go to the website and you will find more information. Our anti-infective pharmacology working group focuses on promoting international exchange of ideas and new research of all aspects of antimicrobial pharmacology. And this is our leadership group with colleagues from US, Europe, I'm from Asia Pacific area. So if you are interested to join our working group, it is free. And simply go to this Google form, spend probably 2 or 3 minutes, and you can finish that. And in particular, in our working group, we have a young investigator committee. This committee facilitates the interaction among young fellows in antimicrobial pharmacology and is co-chaired by Dr. Sunong from Australia and Dr. Amy Huang from US. And we also have 4 very enthusiastic young fellows. So if you are interested to join this young investigator committee, please do indicate in that registration form and feel free to get in touch with Sue or Amy. So our co-host, China Agricultural University, was established in 1905 and is the oldest and best agricultural university in China. It had a huge number of students, and in the latest international ranking, it is ranked the best or the second in agricultural sciences by the leading university ranking agencies. It has a very strong international partnerships with more than 200 universities worldwide. In particular, the College of Veterinary Medicine, it has a very strong research portfolio. In particular, they have 10 national and ministry key laboratories and very well equipped. So at the end of this webinar, you will have the opportunity to ask questions. So for security reasons, you need to go to this reaction button, click the button, and then raise your hand. And one of our moderators will unmute you, and then you can ask questions. So before you ask questions, please introduce yourself briefly, who you are and where you're from. And now it's my great pleasure to introduce our speaker today, Professor Jean-Marc Rolain from France. Professor Rolain is a group leader at the Institute Hospital University Mediterranean Infection, and also at the Pharmacy School of Marseille in France. He's an international leader in molecular biology of antimicrobial resistance and emerging pathogens. He has a remarkable track record with almost 600 publications and H-index 82. And importantly, he's the editor-in-chief of the International Journal of Antimicrobial Agents. So without further ado, I will hand over to Professor Rolain. Now, Professor Rolain, it's your stage.
Thank you very much, Jian Li, to invite me for this webinar. I'm happy to be here with you, even if for me it's very early in the morning in France. So I'm Professor Jean-Marc Rolain, working in Marseille, France, and I'm the editor-in-chief of the International Journal of Antimicrobial Chemotherapy, and I'm an ex-officio member of the society ISAC. So today, I will try to show you a new field of research, and based on the new role of beta-lactamase enzyme, and it's some of the work we have done over the last 2 or 3 years, and some other work are ongoing. And the idea of this webinar is to try to show you that what we call beta-lactamase is not so clear, and what you believe to be beta-lactamase in fact may have other function, other role, firstly, and you will see that you can find now in 2021, based on phylogenetic analysis as well on the structure analysis, that those enzymes are multifunctional and are not only beta-lactamase. So let's move. I do not have any conflict of interest for this talk. So, sorry. Okay, so my talk will start with a short history of discovery of beta-lactam. And it's very important to put this into the concept of One Health. And as Jan said, because you also are linked with the agriculture and veterinary medicine, you will see that there is an importance of this One Health concept, including for beta-lactam and beta-lactamase. Because there is some discrepancies between consumption of antibiotic and emergence of resistance in some remote areas. We'll see some example also of the source and origin of antibiotic resistant gene and the new role of beta-lactamase that we recently proved to be present in human chromosome as well and in archaea that are archaic bacteria that do not have any peptidoglycan, so it's bizarre to have beta-lactamase. So, as you know, it's Sir Alexander Fleming that discovered penicillin on 3 September 1928. I know that it's on 3 September because it's my birthday day. He received the Nobel Prize of Medicine in 45. And it's interesting for those that are researcher, PhD or postdoc, that he discovered penicillin by chance because he was in a conference for 3 or 4 days. And when he came back to his lab, in fact, the culture of staphylococci, you see my mouse or not, Jan?
Yeah, all good, thanks.
Yeah, so he was working on staphylococci here. And then when he came back from the conference, he was upset because there was a contamination of the agar plate with a fungus. And this is the fungus, but he was very curious and said, oh, it's bizarre because my staphylococci are unable to grow in contact with this fungi. Okay, and it's the starting point when he said that this fungus, that was a Penicillium notatum, is able to inhibit the growth of staphylococci. And it's the discovery of penicillin that is here from Penicillium notatum in 1928, but it was manufactured only during World War II because the Penicillium notatum is not so good to produce penicillin, and another penicillin was discovered in the United States by Chain, and that was able to secrete many more penicillin and was the beginning of the story of the human use of antibiotics to treat infectious disease. So beta-lactam antibiotics are most widely produced and used antibacterial drugs in the world, including now in 21st century. And you have many different class of beta-lactam, the penicillin, cephalosporin, carbapenemas, and monobactam. But the thing is that there are all a common structural feature that is called the beta-lactam ring. It's here in red. And then all those compounds in the beta-lactam family contain this ring. And this is what is here in red. And the beta-lactamase are enzymes that are able to hydrolyze the structure of those beta-lactam here for penicillin or for cephalosporin. Okay, so this enzyme can destroy the structure, the beta-lactam ring, and is associated with resistance to antibiotics. So far, this enzyme were only expressed or discovered or described in bacteria. And we call this beta-lactamase, but it was only found in bacteria just because researchers and people like me, that we are stupid, and I'm stupid also, is that we believe that it's just enzyme for defense against beta-lactam produced by antibiotics, by bacteria, sorry. And then nobody look at the possibility that those enzymes may be outside the kingdom of life of bacteria. And the story begins with the concept of One Health. Why? Because what is believed to be the emergence of resistance to antibiotic, including beta-lactamase, previously everybody said, but it was an opinion, that antibiotic resistance in human is associated only with the consumption of antibiotic in clinical and veterinary medicine. It means that the pressure, the selection pressure, is only due to the use of antibiotics by humans, for humans, or for veterinary medicine in agriculture as probiotics. And then you have an impact in the environment because you use antibiotics, so it can go back to the environment, in sewage and the environment. And then the environment could be a source of antibiotics and antibiotic-resistant genes that could be back to human, for example, via food. So the concept initially in the 20th century was that antibiotic usage in human medicine or in veterinary medicine, because it's used also not only for the treatment, but for prophylaxis for animals or as growth promoters. And there are links between these two areas, by food or directly. You have the urban and peri-urban environments that are linked together, or the sewage that are linked together. So everything is linked. And that's the concept of One Health. But there is one thing that is surprising, you will see, is that what about wildlife? Because it has been shown, I will show you some example, that you can find multi-drug resistant bacteria or antibiotic resistant genes in very remote area, for example, in Amazonia, in some village without any use of antibiotics at all. You can find ESBL producer, multi-drug resistant bacteria, in people that do not have access to antibiotics at all. And also in wildlife, I will show you that you can have some multi-drug resistant bacteria and antibiotic resistant genes that are bacteria you can find in human beings, but it's in wildlife, in animals that do not have any contact with human or with antibiotics. So it means that it's not so easy to say that, it's too easy to say that resistance to antibiotics is only linked to human usage. It's not true at all. Now we know that for now with our work for some years. So what could be the one explanation to try to understand why you can have antibiotic resistance gene and multi-drug resistance bacteria in the environment? But in fact, what we know that is mainly antibiotics are produced naturally either by bacteria or by fungi. And in this example, Streptomyces genus is a group of bacteria that is able to synthesize and to secrete many different antibiotics. And those producers may exchange those genes in the soil, in the environment, with non-producers. And this exchange and lateral gene transfer may be confirmed either from soil or environmental bacteria to other pathogens that could be clinically relevant in human medicine. And many different strategies and examples have been shown over the last 25 years by transformation, transduction with integron or transposon. And this is what we call lateral gene transfer. And go back to another thing. I said that the majority of antibiotics that have been discovered over the last century came from natural bacteria found in the environment. And the new concept from last century, the end of last century, is that in fact antibiotic resistance is very ancient phenomenon, because in this first paper published in Nature, Vanessa de Costa showed that you can find by metagenomic analysis in the permafrost in Siberia from samples dating back from 1 million years, that you can find antibiotic resistant genes that are similar to those that we saw in the 20th and 21st century. This is another example of the analysis of the guts of 11th century mummy, where they also found antibiotic resistant genes. And in fact, as you can see here, either from the permafrost here or from this other sample, in fact, you can have here, it's, for example, a mammoth or medieval samples that you have all those class of antibiotics that can be found from very old sample. And then also there is a wide variety of antibiotic resistant genes in the environment with similarity that are between 30 to 85% with contemporary and common antibiotic resistant genes we saw today. This is an example I have done with one of my colleagues here in Marseille. We get a sample that was a medieval bone sample, a human bone sample from a medieval area, 14th century. And from this, we were able, within the metagenome of this sample, to reconstruct a sequence that was a sequence of a metallo-beta-lactamase of 251 amino acids. And then we synthesize this medieval sequence as a protein and express this protein, this enzyme, in E. coli. And we found that it's confer a high level of resistance to beta-lactam. So this is the E. coli, the wild E. coli, and this is, you saw that it's very sensitive to penicillin, tazobactam. And here it's the E. coli with the sequence expressed in this E. coli, and you can say that the E. coli became resistant with a high level of resistance. And so we demonstrate that it is functional. It was the first time that it was proved phenotypically, biologically, that this old sequence, yes, is a beta-lactamase. And this was the first example that we published some years ago. The third thing that is interesting is that everybody believes that the resistance to antibiotic is associated with consumption of antibiotics. And it's this very interesting paper published in the Lancet Planet Health 3 years ago. What was shown is that there was an inverse correlation between antibiotic consumption and antibiotic resistance. It means that the countries where you saw the most important prevalence, including in Asia, of resistance to antibiotic are not those countries that consume a lot of antibiotics. And we get the same thing in Marseille here. So it's a new field to try to explain why you can have such emergence, for example, New Delhi metallo-beta-lactamase that emerged in Asia, why you have such emergence and high prevalence of antibiotic resistance in some place where you do not use antibiotics without any selective pressure. So this is why I have introduced the concept of One Health, because you have microbes in the environment, in the soil or everywhere, that are able to synthesize and to secrete antibiotics, and so they can perhaps select antibiotic-resistant bacteria, antibiotic-resistant genes that came after in clinical medicine. But the only thing we saw previously before the concept of One Health is that it's only in clinical medicine, because we look at this more carefully because of humans. So now go to the beta-lactamase world. So this is a paper we published with my colleague almost 8 years ago. We look at the presence of and the diversity and classification of beta-lactamase enzyme thanks to our database of antibiotic-resistant genes, whose name is Organised. And at that time, around 2,000 beta-lactamase genes were described, with more than 30 groups of different enzymes. But interestingly, if you look at phylogenetically the GC content of all those beta-lactamase, there is a huge difference between some class with some beta-lactamase with GC content as 31 and other as 65. So it means that when you have such a difference in GC content, it means that they came from different source or different bacteria. For example, here you have the common CTX, TEM, and SHV. You see that the GC content runs from 50 to 60%. And you have here the Carbapenemas. And from the Carbapenemas, you can see that KPC is 60%, NDM is 60%, but wow, OXA, it's only 38. So it means that OXA, NDM, and KPC do not have the same origin. And this diversity and classification also is based on the classification of different groups of beta-lactamase. You have two different groups based on functional domain. You have the serine beta-lactamase, with the serine is the cofactor here, and the metallo-beta-lactamase with a zinc that is associated with these two different type of beta-lactamase. And then that's what you know for the classification based on domain, and then based on amino acid sequence. You have from the serine beta-lactamase, we have the class A, C, D with 10 KPC, OXA, and the metallo-beta-lactamase, the class B, such as VIM or NDM. So this is the classification from Karen Bush and Ambler, and this is what everybody is using so far. And if you represent this in a phylogenetic tree, this is what you saw: the class B, the class A, the DD peptidase, and the class D. And it's something that is conserved. And then when, for example, you have a new bacterium, you have the sequence, you can look if you have a suspicion of a beta-lactamase within this tree. You can see if it's a class B, A, or D. But you can do that also with metagenomic and others. And I will show you what is able to try to understand new things. So those metallo-beta-lactamase, the class B, they have a specific motif, a protein motif and the fold that can be found in the sequence, the amino acid sequence of a putative beta-lactamase. And then when we look at that, we found that this motif of metallo-beta-lactamase, you can find this motif in all kingdom of life, in bacteria, in archaea, in eukarya. We just, I will not show you that, but we just have a paper accepted because we also found a metallo-beta-lactamase in giant virus. And then, interestingly, those sequence, amino acid sequence, maybe could have different size, but you can find not only a motif of metallo-beta-lactamase, but other motif with different function, other function. I will show you. So among this class of metallo-beta-lactamase, with more than 34,000 proteins, you can find different scaffold with different motif, with different function. So of course, you can find the scaffold that is associated with beta-lactamase activity, but you can have all those activities. DNA cross-linking repair, RNase activity, glyoxalase, aryl sulfatase, and so on and so on and so on. So it means that those enzymes, in fact, it's what we found now, probably are very, very old, and they have probably a different evolution for a different function if it's in bacteria or in other kingdom of life. So this is the first example of a work on human beings. In our own chromosome, you have 18 human beta-lactamase that have been predicted. So why this, and is it functional? Those of them, so mainly we have worked on this one, MB-LAC1, that is a glyoxalase activity. Here you have other enzymes like SNME 1A and 1B that are believed to have a scaffold that could be a DNA or RNA subfamily activity. And those one are mainly MB-LAC2 that has also other activities predicted by sequence. And interestingly, those two human beta-lactamase also have been shown to be able to rise and to confer resistance to anti-cancer drugs. So it's not only beta-lactamase. So what we have done 3 years ago is to retrieve all the beta-lactamase sequence we get from our Argonaut database. And the idea was to try to build inferred ancestral sequences to look after if we can find this and identify a new beta-lactamase in different kingdom of life. So we construct a phylogenetic tree with all class of beta-lactamase derived from those database of sequence. And we create a common ancestral sequences of bacterial beta-lactamase, 3-1, the 3 nodes here. It means that we call this the mama, the mama of each group of beta-lactamase. And then with those common ancestor sequence from different clade of nodes, we make a blast P analysis to look at possibly existing sequence, but not in bacteria, in archaea and in human chromosome. And then this is the result we get. And you saw all those beta-lactamase with different color. And the conserved motif was present either in bacteria, in archaea, and in eukarya. And then the question is, are those enzymes outside the kingdom of bacteria are able to destroy beta-lactam antibiotics? So we look specifically of these 5 different sequences. This one is a putative beta-lactamase from Metanosarcina, that is an archaea, and the 4 other ones are predictive human beta-lactamase, so in human chromosome. And then we look at those sequences. This is the summary. This is the archaea sequence, and this is the sequence that are putative beta-lactamase found in human chromosome. And then the 3-dimensional structure of those enzymes are clearly associated with a structure of bacterial beta-lactamase, but it's not bacteria, it's human and archaea. And then the two works I will present you now try to demonstrate if it could be beta-lactamase biologically. So the first was the study of those 4 human beta-lactamase, putative beta-lactamase, and try to show that it's effective biologically. So this is the 4 enzymes we look for: MB-LAC-C2, LAC-B2, SNM1A and 1B. And as you can see, 3 out of those 4 human beta-lactamase are able to hydrolyze actively the nitrocephin. You see the MB-LAC2 here, and this one, the LAC2B2 node, is not able to hydrolyze, but the 2 other here, human beta-lactamase, are able to hydrolyze nitrocephin. And then we look at the Cephinase test in human metallo-beta-lactamase in presence and in absence of penicillin inhibitors, penicillase inhibitors, the sulbactam. And as you can see, sulbactam is able to restore the activity and to inhibit the activity of the human metallo-beta-lactamase. This is the human metallo-beta-lactamase alone. And if you add sulbactam, it do not hydrolyze the penicillin. And it's the same here with, here you can see that you have an inhibition when you have sulbactam. So sulbactam is able to inhibit the activity of human metallo-beta-lactamase. And another test we do by LC-MS is to look for to monitor the penicillin degradation of this human metallo-beta-lactamase by quantification of the product of degradation, which is penicilloic acid. And as you can see, this enzyme is able to destroy penicillin, and you have the production of the degradation product. So this human metallo-beta-lactamase is able to degrade penicillin. But now the second question is, okay, it's in vitro model, but is it possible and is it true that it works in human cells? So to finish before human cell, you can see that penicillin G is destroyed by also the two other human metallo-beta-lactamase we predict by sequence. That is the full degradation of the penicillin for the two human metallo-beta-lactamase, and you restore practically the activity if you use beta-lactamase inhibitor, mainly sulbactam. So at the end, you have that 3 of the 4 predicted human metallo-beta-lactamase are clearly metallo-beta-lactamase able to destroy beta-lactams. So now, is it true in human cell? So we use 2 cell culture, the THP1 and MRC5 human cell, and we look if those cells are able to degrade penicillin. And in fact, yes, THP1 and MRC5, sorry, are able to degrade penicillin in the cell and to produce the degradation product. And again, it's restored with sulbactam. So human cell, now it's not only in vitro, but human cell are able to degrade penicillin. And at the end, we try to make an in vivo human cell line models with THP1 infected with pneumococcus. And we use penicillin sensitive pneumococcus. And then what we found is that human cells are able to kill pneumococcus in cell intracellularly. So it means that there is a protection against intracellular infection from pneumococcus if it's susceptible to penicillin by human metallo-beta-lactamase intracellularly in our own cells. You can destroy pneumococcus and it's restored by sulbactam. So it means that sulbactam is able to promote the pneumococcus growth in human cells because it inhibits the beta-lactamase, the human metallo-beta-lactamase activity. And we have also showed this with ampicillin, with degradation of the pneumococcus, and you can restore the activity. And this has been recently published 2 years ago in scientific report, human metallo-beta-lactamase enzyme degrade penicillin. It was the first demonstration of this. The second example is archaea. Archaea are old bacteria that do not possess peptidoglycan. So it's bizarre to predict a beta-lactamase in such class of a kingdom. And then it's a representation of all those metallo-beta-lactamase we predict in genome of archaea and bacteria. And you see we found a lot of them in blue. But what was very interesting is that those blue one, this group of archaea and metallo-beta-lactamase from archaea is grouped with a bacterial group, and this bacterial group is the group of flavobacteria, and it's Elizabeth Kingiae. And the sequence of beta-lactamase we predict in those archaea group with those from Elizabeth Kingiae, whose name is GOB, that is a metallo-beta-lactamase. And as you can see, the motif of those sequences in archaea are clearly very close to those of Elizabeth Kingiae. So again, we look at if it's active. So the Cephinase test of this enzyme, and yes, you can see that this enzyme is able to have activity against Cephinase, and it's restored by sulbactam. So sulbactam inhibits the beta-lactamase activity of an enzyme from archaea. We do the same for penicillin degradation in presence or in absence of penicillinase inhibitor. And again, we found the same as I saw you for human metallo-beta-lactamase. It means that this metallo-beta-lactamase from archaea degrades penicillin, and you restore the activity with the use of sulbactam. We show another thing with those bizarre beta-lactamase from archaea, because we look at imipenem now, because it's a metallo-beta-lactamase that is close to GOB13, and it's an enzyme that is known to be able to degrade imipenem, so carbapenem, not only penicillin. And then we look at this with this sequence MET-B. So you can see that this is the imipenem only. This is the activity of the GOB13. So you can see that there is a degradation of imipenem, and you have the apparition of the imipenoic acid that is the product of degradation. And if you look at the enzyme from archaea, you have the same thing. It means after 1 day, 24 hours, you have a degradation of imipenem and the production of the degradation product. So this archaea enzyme is already imipenem. It has never been shown before that an enzyme from archaea could be a beta-lactamase, and that it's able to destroy also imipenem, the carbapenem group, even if some people say that beta-lactamase, but it was 25 years ago, beta-lactamase are absent from archaea. It's not true now. And this is what you can say if you look at the antibiotic susceptibility testing of those bacteria, those archaea, they are very resistant to antibiotic, including to imipenem. So that's raised the question why archaea have active and biologically active beta-lactamase, because they do not have peptidoglycan. What is the role of this enzyme from this archaea? Perhaps it's for protection against imipenem producer in the environment, probably. And I will just show you one paper we recently published, because imipenem, the first discovery of the carbapenem group was shown in Streptomyces cattleya, that is able, thanks to a non-ribosomal peptide synthetase cluster, to secrete tianamycin in the environment. So probably archaea are able to survive in this different ecosystem because they are able to survive to the attack of bacteria producing carbapenem. And then from this archaea, we also found that the sequence also have a scaffold predictive to be perhaps nuclease or ribonuclease. So we look at this activity. So this enzyme for DNA and DNase activity, we do not find any DNase activity. But for RNase activity, you can see that there is a significant RNase activity of this enzyme. So it's not only a beta-lactamase, it's also an RNase. So it's probably very important for the archaea to have this enzyme also to destroy RNA and outside or foreign RNA. So it's a defense system. So we also published this in Life the dual function of RNase and beta-lactamase activity of this very, very strange enzyme. So this is another paper we published in Scientific Report showing that Streptomyces cattleya, as I said, is the bacterium in actinobacteria, is the bacterium that is able naturally to secrete chenamycin, that is an analog of imipenem. And what we found in this cluster of genes that is able to synthesize chenamycin, so a carbapenem compound, there is 2 genes in this cluster that in fact are carbapenemas. So we found that one gene whose name is THNS, that is in this cluster, is the antidote of chenamycin. It means the bacterium secretes chenamycin, but to be able to survive against chenamycin, in this cluster, the bacterium is also able to synthesize a carbapenemase so that the bacterium can survive by itself because the bacterium has the antidote. And some other example of bizarre things is that we published that you can find antibiotic resistant genes and carbapenemas in very, very remote area, including in wildlife. So in this first paper, we discover a huge prevalence of OXA48 carbapenemas in wild boars in Algeria. It's in the wildlife because they are not in contact with antibiotics. And again, we also find this in a small monkey, the Barbary macaque in Algeria with CTM15. And then you can find a huge amount and prevalence in those arrays. Another thing that is very bizarre, we have published this some years ago, about 10 years ago, is that we found sequence of carbapenemas that are likely close to Acinetobacter baumannii, OXA23 carbapenemas, and we found this in human body lice in Senegal. So it's very bizarre, and we screen human head lice, human fecund sample, and animal sample, and we found this. So you can have also reservoir of carbapenemas in arthropods. We have recently published also this work showing that we can find multi-drug resistant Klebsiella clones from wild chimpanzee and termites in Senegal, in place where these chimpanzee and termites are outside human place, without any contact with humans and without any use of antibiotics. So those, interestingly, those Klebsiella, you can find them in the termites that are food for chimpanzee, and the clone of Klebsiella pneumoniae we found in those termites and chimpanzee are ST307 and ST147. And for those that are working in clinical medicine, you know that those 2 ST clones have emerged recently in clinical medicine, but it's also present in chimpanzee and termites in Africa. And finally, this is also a very bizarre thing and interesting. People have published a paper showing that a small arthropod, this one, that is commonly called Colombola, whose name in Latin is Folsomia candida. So it's a small arthropod. And when they look at the genome of this small arthropod, they found that there was a full cluster, a non-ribosomal peptide synthetase cluster, that is a cluster able to synthesize penicillin. So it means that you have the full cluster that has been transferred, probably from a fungi to this small arthropod. It's in the chromosome of this arthropod and can synthesize penicillin. So why do arthropods secrete beta-lactam? Probably again, it's a difference against penicillin susceptible bacteria that can perhaps kill the microbiota of those small arthropods. So take home message, something that you should have in mind is that beta-lactamase enzymes are not only present in bacteria. You can find this everywhere, archaea, eukarya, giant virus. And if you express those enzymes, yes, they have a penicillinase activity, including those in human. And for archaea, we also show that there are other roles such as RNAs or glyoxylase. And interestingly, there is another thing that is another putative role of that is that some bacteria are able to use antibiotics as carbon source to grow. So it could be also a role of antibiotics in the environment. And then, of course, we need to qualify the annotated enzyme because there is not only beta-lactamase activity in those sequences. So in conclusion, I hope I open a new field for you because it's a new field to be explored for me, that there are many other roles of beta-lactamase in different kingdom of life. And it remains poorly understood at that time. So there is a lot of work for researchers on this topic. And then those questions that why beta-lactamase are found in remote area and these different ecosystems without any antibiotic pressure. And perhaps there is another pressure. What we have shown and published recently is that probably some insecticide and pesticides such as glyphosate may be a selective pressure for antibiotic resistance as well. And again, why you can have arthropods that secrete beta-lactam. So this is time to close this this talk and to open the question. Thank you very much.
Thank you very much, Professor Rolain. That's a fantastic talk. I'm sure there's some questions in the audience. So I also have some, but I'll probably leave to the audience first. So any questions from the audience? So maybe while we are waiting for them, I just ask a couple questions here, Jean-Marc. In your study, you use MRC5 and THP1 cell to show the degradation of the beta-lactams. I just wonder, have you knocked out these two particular, a few particular potential beta-lactamase in the mammalian cell and then see whether that degradation disappears?
No, we have not done this, but because it's difficult to do that because you need to be able to use for example, the CRISPR-Cas9 technology, and at that time we are unable to do that in the lab. I don't know how to knock out by CRISPR-Cas9 gene from human cells.
Yeah, I think now probably a little bit easier, but I think this is a really interesting phenomenon. And also, as you mentioned, why even animals they produce beta-lactams and humans. So do you think one of the reasons could be, beta-lactams really exist widely in the environment, and for whatever reason, maybe mammalian cells also need to respond to that, or they by some non-specific mechanisms which just degrade beta-lactams for some other reasons?
What I think is that it's only produced in specific ecosystem. Okay, it's the theory of the group of bacteria, archaea, fungi in a specific ecosystem, but the amount of antibiotic that are produced in this ecosystem is very low as compared to the use of human of antibiotics by humans in medicine or in veterinary. So that's why we are looking for other selective pressure that can explain the emergence of resistance. And then we published that in the JAC this year, that there are no evidence that pesticides such as glyphosate may be a driver of selection also of antibiotic resistance, because the company Monsanto, when they patented glyphosate 25 years ago, it was patented for use in plants as a pesticide, okay? But 10 years ago, Monsanto, they also have a second patent of glyphosate to say that glyphosate is an antibiotic. And glyphosate has antibiotic activity against bacteria, but has also activity against some fungi and apicomplexa, such as Leishmania or Plasmodium. So it means that what we call, for example, pesticide, it's because it's used as pesticide, but it's also an antibiotic. It's like when you say it's semantic. If you said anti-tuberculous agents, it's against tuberculosis, but anti-tuberculous agents are antibiotics as well. So it's just a semantic. And another example is that for antiviral drugs, you say it's against viruses. But myself, I demonstrate and I publish that zidovudin, AZT, the first anti-HIV drug, is able to kill every E. coli, including multi-drug resistant E. coli. So it means that we said it's an antiviral drug, but it's also an antibiotic. So it's just the semantic at the beginning. So we need to make new classification because you can make drug repurposing using this mind.
Yes, absolutely. I just saw Sue send in the chat. So if anyone doesn't have access to a mic, you can send your question to the email. So please feel to do so. Any other questions from the audience? I'll probably ask another one, Jean-Marc. So in your bioinformatic analysis, did you see any particular beta-lactams exist in plants, for example, compared to mammalian cells?
Yes, in fact, the thing is that those enzymes that are very probably very ancestral have a different evolution. If they are in bacteria, the evolution was to optimize the beta-lactamase activity. And for example, in human cells, the evolution of those sequences make this enzyme more able to have an herein activity. It means that according to the group, the plants, the bacteria, or eukarya, the evolution of this enzyme are adapted to the ecosystem and to a different role. It means that the human metallo-beta-lactamase have a small beta-lactamase activity. It works. We have demonstrated this, but the herein activity is much higher because the evolution of the protein was adapted to the ecosystem.
Yeah, I think, certainly nature mother is very smart. I don't think, no matter any kind of organism, they make this enzyme for nothing. So there must be really good reason. This is a very, very interesting question.
Yeah, it's the theory. It's the theory of Ernst Mayer's for the gene. It's use it or lose it. So if it has not been lost in human or in plants or whatever, it means that there is probably an explanation. There is a role of those enzymes because they have not been lost during the evolution.
Yes, absolutely. So we only have 2 minutes left. So are there any other questions from the audience before I close up? I guess this is a format. I think people can easily raise your hand and then we can discuss this. All right, if there are no more questions, before I finish up, I would like to thank firstly, Professor Jean-Marc Rolain for the fantastic talk. I know it's very early morning for you. I really appreciate your efforts for this webinar. And also I would like to thank the tremendous work behind, in preparing this webinar. For example, Sue, Fakade, Amy, and John, and Clay, they all put a lot of effort into this. So I do encourage young fellows, if you are really keen to do some networking, and please join our Young Investigator Committee of the working group. And finally, I would like to thank Professor Jihui Hou and Pingping Wang from China Agricultural University, and a lot of hard work they put in behind the scene as well. And thank you for attending this webinar, and I hope everyone have a good day or have a good evening, depending on where you are. Thank you all.
Thank you, John. Bye-bye.
Take care. Bye-bye.